So it gives me great pleasure to introduce my friend and colleague Lee Hood. I think for many of you, Lee requires no introduction, but I'll subject him to a short introduction anyway. Lee is a very untypical person in being quite a visionary, the kind of visionary that comes along very rarely. Most of us say that it's hard to predict things, especially about the future. Lee has probably been more correct about predicting things farther into the future than most scientists in the history of science. And not only predict, but then put together methods and technologies to deliver on his concepts of how science would move forward. He's a leading scientist in molecular techniques and genomics. Lee, I won't divulge his age, but let's just say that he was 10 years ahead of his time when I was born. He obtained his MD from Johns Hopkins University in 1964 after being an undergraduate at Caltech. Then went back for a PhD, sort of a double, not a combined MD PhD, but two sequential degrees, which in the US is actually quite uncommon. Obtained his PhD in 1968. Has well over 600 papers, very significant papers in the literature. He pioneered DNA sequencing, the DNA synthesizer, protein synthesizers, and protein sequencing. He's most well known for the sequencer, which led to the data deluge that most of us now benefit from in terms of the huge amount of genomic information. He's had many prizes, all of the typical prizes of outstanding scientists. Here in Japan, most significantly, in 2002, he was awarded the Kyoto Prize. He's now moved, I guess for the second time in his career, to couple his talents and interests to things related to neuroscience, and he'll tell us a little bit about that. But some of you may remember at some point in the past, someone came up with a way of introducing a gene to solve an animal model that was screwed up, the shiverer. That was an early transgenic activity that Lee had carried out. So, Professor Hood.
Well, thank you very much, Mark. What I'd like to do is talk both about systems approaches to studying the brain and to studying medicine. And at the very end, I'll give you just an indication of the revolution I think is coming in medicine. And it isn't an incremental change, it's a revolutionary change. Medicine is going to be information-based, and my vision in 10 years is that we'll see each patient surrounded by a virtual cloud of billions of data points, and we'll have the IT for healthcare to reduce that data dimensionality to simple hypotheses about health and disease. What this means, of course, is that medicine really is becoming an informational science. And I'd like to begin by talking about just how, at least I think about biology and medicine as informational sciences. I'd say, one, that there are two very fundamental types of biological information. One is the digital information in the genome, and of course the second is the environmental information that impinges upon the genome. And in fact, the whole purveyance of systems biology is how you integrate these two types of information together to create phenotype. Phenotype for development, phenotype for physiologic responses or disease response. One of the interesting challenges is how do we collect that collision of two types of information with the phenotype we see. And of course that is mediated by two general categories of informational structures. The first, of course, are biological networks that will capture and transmit and integrate and modulate information. And of course they hand it off to molecular machines that actually execute that information. So again, understanding both the spatial and temporal dynamics of biological networks and molecular machines is the essence of systems biology. Now, if you ask the question, how do I view systems biology? I view it in several different contexts. First, I think it has to exist in a framework that's cross-disciplinary in nature, that brings together all of the major classes of scientists. And the essence of how I view this framework is this dictum that it is biology that has to drive the type of technology you develop, and that in turn will lead to the development of computational and mathematical tools. So biology is always the real driver of systems biology. And what this means is you have to coordinate the activities of these different scientists together to attack big integrative kinds of problems. And that requires two things. One, they learn to speak the language of one another so they can communicate effectively. And two, they work effectively together as teams. And neither of those is particularly easy to accomplish. What I think has really been important for ISB is we have both the information technology integrated to the computational mathematical tools, and those can feed directly into simple and complex models, ultimately into human disease. And at ISB, any scientist has access to these tools for developing new types of ideas. What, of course, is the essence of systems biology, I would argue, is that it is hypothesis-driven. You look at a complex system, you take all of the information you have, you formulate a model to explain it, and then you create hypotheses to test that model. You carry out perturbations, you integrate the information back into the model. So in an iterative way, you bring the theory into apposition with the experimental data. Systems biology has three important things to say about data. One, you should accumulate global data, comprehensive data, all genes, all transcripts, all microRNAs, what are they doing? All proteomics, but that's more limited in technology and so forth. Number two, you want to be able to integrate the different scales of data from DNA to RNA to protein to networks and all the way on up to organisms. And it's the reason for this integration is that at each of these levels of information, the environment impinges upon that core digital signal and modifies it. And what we mean by the integration of different types of information is nothing more than the explication of what the environmental contributions successively have been. How to do that is one of the grand challenges of systems biology. We want to delineate the dynamics of biological networks, and that's both temporal dynamics and spatial dynamics, and I'm going to talk about both of those dynamics a little bit later in this lecture. And ultimately, what we'd like to do is come up with models that are predictive and actionable. That is, they formulate predictions that can be tested, and if you're a physician, they give you hypotheses that are useful in guiding the patient to where to go. And it goes without saying that discovery science is really a key part of systems biology. And by discovery science, I mean the enumeration of biological objects. And the Genome Project was the prototypical classic example of discovery science. And I'll mention very briefly about the Human Proteome Project, another project that's coming up that will be similar at that level of information. On a personal note, ISB is now 10 years old. I started ISB in 2000, and we've grown to a staff of about 300 with 13 faculty members. And you're always interested at annual birthdays about the metrics of how successful you've been, and not only the metrics of how successful you've been as an institution, but how successful systems biology has been as a whole. And about 4 months ago, we came across a wonderful study by a Spanish institute that develop new metrics for assessing the impact of papers, and then being able to average all the papers across a given research institute to come up with a metric score of contributions. They tested this against 2,200 research institutions in all disciplines, and they came to the conclusion, as far as ISB is concerned, that in terms of impact, we were first in the US and third in the world. And the high impact papers we generated were in biology, were in medicine, were in computation and mathematics, and were in technology. And it was an enormous validation of the power and the momentum, I think, that systems biology has gained. The systems view of disease, of course, is the idea that disease arises as a consequence of networks that become disease-perturbed, either genetically and or environmentally, that these disease-perturbed networks alter the envelope of information that they express. They do so dynamically, and it's this altered, dynamically changing envelope of information that encodes both pathophysiology and gives us new insights into diagnosis, therapy, and ultimately into prevention. And I'm going to talk about two kinds of studies today. One are temporal analyses that we've done on the transcriptome in the brain of the mouse during progression of Prion disease. And the second are spatial analyses we've done in imaging cell type specific transcript in the mouse brain. And they both illustrate in beautiful ways the power of, again, dynamic analyses temporally and spatially. And of course, ideally, we'd like to be able to integrate these two types of studies together. And as Mark can tell you, that is incredibly difficult. So I'm going to talk about Prion disease in mice, and we're going to be interested in what are the disease-perturbed networks that exist there. As many of you know, Prion disease arises as a consequence of a protein whose structure has been altered, and when it's altered, it acquires two interesting properties. One, it can catalyze the conversion of normal prions to their infectious disease-causing form, and two, the infectious prions are generally resistant to virtually all known proteases, and maybe this in part adds to the aggregation that occurs in neurons that leads to their death and the pathophysiology of the disease. The approach that we took was to use inbred strains of mice to infect one set of inbred mice with prions in the thalamus, and then follow the disease over the course of progression. C57 mouse is about 22 weeks of progression, and at the same time follow controls and look at 10 different time points, analyzing brain transcriptomes and other things that we'll talk about during this temporal time study. We ended up doing 8 prion combination, inbred strain combination of animals in this way, for reasons I'll talk about in just a moment. And it had to do with the fact, in looking at these 8 animals, 7,400 genes were differentially expressed in one or more of the strains in diseased as compared with their normal controls, a subtractive analysis revealing information. And of course, the essence of what that says is, in these 7,400 genes we're looking at, there's an enormous amount of noise. So how do we extract signal from the noise? And the essence of really what we wanted to do in this process was to define a number of genes that specifically encode the degenerative neurological process of Prion disease. And I'll just tell you that there are 2 sources of noise that are enormously important. One is technical, and I'm not going to talk about that. We can deal with that with statistics and lots of other things. But the other is biological, because we realize when you assay any particular phenotypic aspect, you integrate together many different interesting biologies in that process. And if you're interested in only one of them, you have to be able to subtract that one away from all the others that contribute to the signal. And the approach that we took for doing this was to use these 8 Prion strain, inbred strain combinations, and I'm not going to go into the details, except to give you some examples of how we can use subtractive analyses to subtract biological noise from the core prion response genes that we're really interested in. So one example was one of these animals was a double knockout for the prion gene. When infected with infectious prions, they never get the disease. Any changes in that brain transcriptome are irrelevant to the neurodegenerative process, so we could subtract those away. And we did 8 other subtractions that were very, very parallel. And in fact, we were able to reduce this 7,400 genes down to a core of about 333 genes, which we think encoded the essence of this prion response. There have been 30 years worth of beautiful histopathology done in the mouse, more recently in human, that have defined 4 major types of processes that are going on during prion progression. One is prion accumulation and replication, glial activation, and then 2 aspects of neural degeneration, the degeneration of the extremities, and finally apoptosis, the nerve cell death, and so forth. We can go to the literature and we can define protein interaction networks that mediate at least many of the processes that are involved in these endeavors. And so what we've done is integrated the 333 genes that we think are the core from the brain transcriptome together with the protein interaction data from these 4 different networks. We also did dynamic histopathologic analyses at these 10 different time points so we could do change correlations with phenotype as well as following the clinical signs. And we did sagittal sections of the animal to follow, in a temporal sense, the distribution of the spreading infectious prion protein. And finally, and I'll talk a little bit about this later, we have brain-specific blood proteins that have made remarkable revelations about the nature of the pathology that was going on in the brain. So what I'm going to show you are 3 panels from the neural progression of Prion disease in C57 black, a disease that progresses to termination at 22 weeks. And I'll give you a slice at 6, 10, and 20 weeks of progression. In the upper left-hand corner is a little box that says there are no clinical signs at 6 weeks. And in the lower right-hand corner is the sagittal staining for infectious prion. There's not much there either. And yellow indicates that the normal transcriptome and control transcriptomes are essentially identical one to another. If you look at 10 weeks, you begin to see changes, the elevation in the diseased animals of certain transcripts, and we'll talk about the nature of those transcripts a little bit later on. No clinical signs yet, and the beginning of the revelation of infectious prions. At 18 weeks, the very first clinical signs come. At 22 weeks, they're manifest. You can see massive spreading of the infectious prion and massive changes in the nature of various aspects, the subnetworks that together encode prion accumulation and the replication network. What was interesting about these 4 networks is that they were sequentially activated during the disease process. Prion accumulation was first. It is the unique network to this whole process. And the next 3 actually are common phenomena seen in other neurodegenerative processes. You can look at humans and see similar kinds of events going on in human neurodegenerative disease. But there was a sequential activation. This has really important implications for drug targets. If you want to hit prion disease early, you obviously ought to have drugs that could be pointed at the prion accumulation network so that you can change its behavior from disease perturbed to more normal, and to block its activation of the effector downstream networks and so forth. And the same is true for diagnosis. If you want early diagnosis, you want to look at changes that are occurring in this first network. One of the big reservations we had about carrying out the studies in whole animals was whether the signal to noise could be seen in the whole brain, and it could very nicely. But the other issue was, could we make the correct neuronal cell type assignments for the various transcripts? And so we took 128 of our key transcripts and compared them against the Allen Brain Institute in situ hybridization data, and we were able to show that we made the right calls in these 4 networks every single time. So it was mute testimony to the power of this approach. So we can draw 5 major conclusions. One, there is this core of 333 genes for prion disease. 2/3 of them encode these 4 known disease-perturbed networks that come out of the histopathology, but the remaining third encode 6 other networks that no one had any idea had anything to do with prion disease, in so-called dark genes of prion disease. The sequential perturbation of the 4 major networks I've commented on, and of course the idea that you can use drugs to re-engineer networks to make them behave in a more normal fashion is going to be really a powerful new approach to drug target discovery in the future. We think we can use systems thinking in blood diagnostics to make blood a window into health and disease by using these same systems approaches to thinking about blood diagnostics, and there are 2 aspects to it. One is we can, from the network, get biologically relevant proteins that can mark, say, the early onset of a disease, and if they're secreted into the blood, we can make early predictive comments about it. But even more important, I think, is the fact that we've taken on the task of identifying organ-specific transcripts that are specific signposts for what's happening in the brain. So here are 3 panels from the prion accumulation network. You can see at 12 weeks, 6 weeks before there are any clinical signs, a number of transcripts have changed, and we were able to show 3 of those transcripts were in fact secreted into the blood, and all 3 had elevated levels in the blood that gave us the ability to do pre-symptomatic diagnosis. But there is a fatal flaw in doing biology alone, and that is all 3 of those transcripts were encoded in multiple organs, and a priori we wouldn't necessarily be able to say these only came from the brain. So the second approach was to use deep transcriptome analyses of 40 different organs in humans and mice to identify relatively organ-specific, brain-specific transcripts, and there turned out to be more of these for more organs than we'd ever thought. What is interesting about these brain-specific proteins is in the blood they constitute a molecular fingerprint, and if you look at the proteins in the fingerprint, in a normal brain they'll be at 1 level of concentration. In a disease brain, those proteins whose cognate networks have been altered will change their levels in a very specific pattern because different diseases alter different combinations of biological networks. So you get pathognomonic characteristics that will allow us to separate normal brains from disease brains, and if disease, to identify which disease one would have. We've gone on to look at these transcripts, and we now have, for the mouse brain and essentially for the human as well, more than 100 brain-specific blood proteins, and for liver, which we're doing in other studies we won't talk about, more than 80 of these, and we've made these all specific MRM assays of enormous sensitivity, so we have the ability then to interrogate brain behavior, and I'll tell you a little bit later about how we're going to use those in very interesting ways. We did take 15 of these brain-specific proteins and demonstrate that in 3s or 4s, they were encoded by each of the 4 major networks that we defined earlier. And what we were able to show beautifully is in the blood, we could distinguish the early disease-perturbed networks from the late disease-perturbed networks. So that means from the blood alone, we can begin to make assessments about disease progression. And one final point that I'll make is I think microRNAs are going to be an unbelievable diagnostic tool for the blood. And here we show you a series of microRNAs that are elevated and another series that are decreased during the progression of prion disease. And I'll point out that these changes occur well before we can see any changes in the proteins themselves. MicroRNAs in the blood are incredibly stable and infinitely easier to diagnose than our proteins. They'll become a very useful marker in the future. So our view is by using this combination of biological features and organ-specific features that we'll be able to do early detection, we'll be able to do disease stratification, and that's really going to be important for the impedance match against the right drugs in the future. We'll say more about that later. We're going to be able to follow disease progression, follow therapy, and address reoccurrences. And I'll say in full disclosure, we started about 9 months ago a company called Integrated Diagnostics that is basically using these strategies for blood diagnostics. And if it goes well, we think that will be a very powerful platform company for this thing we call P4 medicine. So let me talk about a collaboration with Nathan Price at the University of Illinois to look at the spatial organization of cell type specific transcripts in the brain. You all know that the Brain Atlas has done a marvelous job in being able to look at most mouse genes with in situ hybridization down to the single cell resolution. So one can map in enormous detail transcriptomes that are present there. We were interested in using these data to do three things. One, to look at those transcripts which we'd shown were brain specific. Number two, we started looking at transcripts that were brain subregion specific, and we divided the brain for purposes of this analysis into about 14 different subregions. And more recently, we've been looking at cell type specific transcripts, and that's what I'm going to talk about. And the idea here, of course, is to take major organs like the brain or the heart or lungs, liver, and to create these various classes of transcripts and to see whether they're secreted in the protein. And what we were particularly interested in is looking at myelinating cells, oligodendrocytes, looking at neurons and looking at astrocytes to see if we could get the brain-specific transcripts there and to see how those brain-specific transcripts would end up mapping into the brain. A study was done recently that essentially used cell sorting to get the three major classes of brain cell types, and then deep transcriptome analysis let one get neuron-specific, oligodendrocyte-specific, and astrocyte-specific transcripts. And for example, there were 367 neuron-specific transcripts and lesser numbers of the other cell type-specific transcripts. And the idea, of course, that the Allen Institute had already shown is if you take specific neuronal specific proteins, you can generate beautiful patterns of regional specificity, showing that there are distinct expression patterns in the brain. And what we wanted to do is to take this analysis and to look simultaneously at 367 neuron-specific transcripts and see how they mapped in ways that I'll talk about in just a few moments. So we looked both at coronal sections and at sagittal sections. We looked at major regions in the brain here, and we were able to essentially take each of those slices, divide them into 10,000 pixels, and then for each pixel create a 367 dimension vector that in a sense was a fingerprint for that pixel. And then what we did was use a procedure called k-means clustering to say, in accordance with these integrative vectors in these 10,000 pixels, can you divide all of those pixels into 3, into 4, into 5, into 6 different groups? What would happen if we forced the classification using that 367 dimension vector for the neurons? And of course, the results really turned out to be interesting. So with a k-means cluster of 3, that is, you're asking it to do 3 classifications, you can see here that you have features that are well known in brain anatomy. When we did a k-means cluster of 4, you got additional specificity. With 5, you got more specificity. 6, 7, and 8, you increased the specificity absolutely beautifully. And in fact, we've taken it up to into the 30s, and there is a point of saturation once you get in the 20s. But the really interesting question is, what are the distinct region-specific, neuronal-specific transcripts that define each of these regions and define them successively? And we'll talk more about what that means. If you look at the sagittal sections, you see exactly the same kinds of thing with a clustering of 3, of 4, of 5, or of 6. You see increased specificity of definition of the features of the brain, and going on up to 10, you can continue to do this. So there's a wonderful correlation between gene expression and temporal features of the brain. And although it's less convincing, exactly the same is true for oligodendrocytes and for astrocytes. The boundaries aren't nearly as sharp, and the specificity, the regional specificity, is not nearly as complicated. But the implications of this are really staggering. Can we identify subtle region and cell type specific transcripts that define different regions? And in fact, ultimately, some of these will be secreted into the blood, so we can specifically pinpoint the activity of certain kinds of regions. Can we understand the developmental process in the brain in terms of this specification of the increased cluster and organization of the brain in this sequential process? Are these things correlated in interesting ways with one another? Can we delineate different major types of neurons? And the question of how many there are, it's a question that has fascinated neurobiologists forever. And can we correlate the 3 cell type specificities in 3 dimensions with one another to see what types of specificities correlate one with another? And of course, you can get into structure function problems and a lot of other kinds of possibilities. And we think this has enormously important implications. And if you could integrate the spatial with the temporal kinds of changes, you really have something interesting. Let me talk a little bit about strategies and new kinds of technologies. We recently published a paper in Science where we did whole genome sequencing of a family with a company called Complete Genomics, who did a superb job in the sequence analysis. We looked initially at a family of 4. The parents were normal. The kids each had 2 different genetic diseases. In sequencing these complete genomes, we were able to show, one, that using the principles of Mendelian genetics, you can correct more than 70% of the normal DNA sequencing errors, generating an error rate of far less than 1 in 100,000. And the accuracy of these data led us to really interesting things I'll talk about in just a moment. Number 2, we can, within the family, discover rare variants. We discovered in this family 230,000 rare variants because you can confirm it's a rare variant, not a sequencing error, if 2 or more members of the family has that same rare variation. And we think these are really important because we think they're going to be the substrate of most disease-causing genes. And I'll make arguments for that a little bit later on. One of the things we were able to do is, using Mendelian principles, is specify a detailed recombinational map of the 2 children, showing how they put together various combinations of their parental haplotypes. We showed that the ratio of male to female crossing over, roughly 2 to 1, favoring females, is what was theoretically expected. What was a little surprising is 65% of the recombinational sites were hotspots of recombination. That obviously has important implications. For the first time ever in humans, we were able to measure intergenerational mutation rate by taking 60,000 sites that varied, where they could be sequence errors or they could be mutations. We used hybridization selection from adjuvant to re-isolate those 60,000 sites, re-sequence them, and we got an error rate of about 10 to the minus 8. That's roughly 30 mutations per child per generation. And the interesting question is, will different families have the same mutation rates or very different mutation rates? And we're well into the study of a whole series of other families to answer that question now. But what was most interesting is you could set up for these simple Mendelian traits either simple models or compound heterozygous models, and then you could model these formulations if you had the information from one individual, from any two individuals, from any three, or from the whole family. And what you showed in all cases was there was an exponential decline in the number of disease gene target candidates. And ultimately, with the correct model, we came up with 4 candidates out of 20-some thousand genes. Chromosome 4, chromosome 5, and 2 on chromosome 6, and it was relatively straightforward to make the assignment to the 2 different diseases. So my contention is now that any simple Mendelian trait is mappable in a straightforward way. We're now working in Huntington's disease, 65 individuals from families to look for modifiers of disease. And I'm going to tell you in a moment how we think we can use family genome sequencing to really attack complex genetic diseases. And you can only do that after appropriate stratification. And Alzheimer's disease is one that we will be working on. But you need to know one more fact before I get there. And that is we can now do single-cell analyses that are really transformational. Study we did about 2 or 3 months ago was to look at a human glioblastoma cell line and to analyze 32 cells quantitatively for 24 different transcripts, most of them CD markers, so we could do cell sorting. And we were able to show that we could divide those 32 cells into 3 quantized populations, that is at least 30 of the 32 cells, all tightly clustered and each quite far away from one another. So what the meaning of those populations is, we don't know, but we're in the process of doing cell sorting to separate them, be able to study those populations in enormous detail. But I think single-cell analysis is going to be one of the really important new transforming technologies in the future. And in fact, this gives you a hint of how you ought to carry about the Cancer Atlas project, because now they're sequencing heterogeneous cells from a single tumor. And if you did that in this particular case, you'd average out the signal, you'd enhance the noise, you'd get exactly the inverse of what you want. And that isn't to say the Cancer Atlas project hasn't been interesting, but it's missing, I think, the most important data that's going to come out of cancer biology. We have 3 big new projects at ISB, and I'll talk about the third one very briefly. The Human Proteome Project, we're going to use MRM mass spectrometry to generate assays for all human proteins. We think we can do that in the next 2 years. But we're putting together a program for federal funding agencies that really parallels the Human Genome Project. Number 2, we'd like to set up in Seattle the tools to be able to obtain all complete human genome sequences and their phenotypic and/or clinical data so that we can mine those sequences and their information for the predictive medicine of the future. And the third project is clinical assays for patients that allow new dimensions of data space to be explored. And I'm just going to give you an indication of some of the things we're doing. For genomic assays, there are 6 or 7 that we're working on. The complete individual human genome sequencing of families, obviously, is one of those. We have a whole series of assays in proteomics, and I'd be glad to talk with any of you about those as well. And we have assays that are analyzing single cells, and we're particularly going to be interested in blood cells because I think either those of innate or adaptive immunity are going to turn out to be enormously informative. And we're particularly excited about iPS cells. What our collaborator, Cellular Dynamics stem cell company in Madison, Wisconsin, has done is show from a mill of blood you can easily get iPS cells for the individual. And they're working on differentiation procedures for taking those iPS cells to neuronal cells in enormously high yield, and that should be done in just a few months. And we'll talk about what we're going to do with that right now. So how do we think about complex genetic diseases like Alzheimer's? So the idea would be to collect the families of patients with the relevant disease, to create iPS cells from each individual in these families, to differentiate the iPS cells to neurons and test tubes for each of the members of the family, and then to probe the individual neurons at the single cell level with transcriptome analyses to identify the extent and degree of heterogeneity of those neurons, and if necessary, sort them into different populations. Then we would interrogate these neurons with natural ligands, with drugs, relevant microRNAs, and analyze then their transcriptomes and microRNAomes and proteomes with regard to multidimensional response to perturbations. And this is the essence of how we think we can do the stratification. And then we will do a global comparison across families and see if we can't end up stratifying. And these studies will be underway over the next couple of years. The idea that we can bring together then a combination of techniques with systems approaches, with new kinds of technologies, with new computational and mathematical tools, leads to this predictive, preventive, personalized, and participatory medicine. And I've defined the essence of it for you. It's going to be information-driven kind of medicine. It's going to be a highly personalized kind of medicine. And of course, it represents an enormous challenge, so we've set up 2 strategic partners initially to attack these challenges. So to develop the tools and strategies for P4 medicine in the patient assays, we have a collaboration with the state of Luxembourg for about $100 million over the next 5 years to be able to take this project on in an enormously big fashion. And then more recently, we've set up the P4 Medicine Institute with Ohio State Medical School. And the idea here is they are payer provider for their 55,000 employees. We have 2 pilot projects for which we'll be using these 55,000 employees to begin applying various of the assays that I talked about before. So in closing, let me just give you the dimensions of how we see P4 medicine. It is the medicine of the present and the future. It's driven by systems approaches. We are going to be able to develop metrics in the not too distant future for measuring, quantifying wellness. We think it's revolutionary rather than evolutionary. It's patient and or consumer driven. The pilot projects will be essential to convincing skeptical physicians about the dimensions of opportunities in this new area, and it's going to lead to, I think, a transformation in the healthcare sector. Every one of the companies will have to rewrite their business plans in major ways, and that will present new opportunities, obviously. And I think it's going to be effective, and I think it's going to be less expensive. I think we'll be exporting this to the developing world in the near future. And I hope, in fact, if we go around another debate on healthcare, that it can be framed around P4 medicine rather than around the failed reactive medicine of the past. Thank you.
So, Lee, thank you very much for that stimulating, very exciting talk. I think we have a question.
Let me add my thanks, Lee, and it's great to see your vision and energy harnessed to neuroscience-related issues to the degree that it's now happened. The question of many that I wanted to get your inputs on is the relationship for these various brain-specific blood proteins. What's known at this stage about how changes in the blood relate to changes in within the brain parenchyma proper, and because I assume these proteins are getting across the blood-brain barrier in various ways that may not yet be well understood. So what can you say about it at this stage?
So I can say two things. One, we were surprised how many proteins got across and into the blood. I mean, we've been able to find, I was hoping we'd get 10 or 15, and we've got way more than 100 now. And it's just
and this
a matter
is in
of
healthy animals? This is not disease-related necessarily? Or?
So that's a very complicated kind of question, and it has to do with how complicated the blood is as a proteomics complex. So to be able to see many of those proteins, we have to reduce the front-end complexity of the blood enormously. But we've been able to see most of the things that we talked about in that 110, one way or another. In some cases, there is very little that's seen in the normal, and there are very high levels in the disease. In other cases, it's the inverse, so there's no easy way to predict. But I will say that the secreted proteins, interestingly, come from three sources. One, they are classically secreted. Two, they are cell surface proteins that are cleaved by proteases, and disease often activates proteases. And three, they're internal proteins that are released by cell lysis through death and things like that. So we see proteins in all of those categories. In some cases, I would say in the majority of cases where we've looked at it, the change of level we see in the protein reflects the change of level we see in the brain and the transcript. We haven't done brain proteomics to know whether the brain proteins are changing or not. So that's kind of the state of the art.
Right.
Try the next one up. It's on? Yes.
That's not on.
Hi. Okay, that works. Hi, I used to be a wet lab proteomics guy, and I just have a quick question for you. Considering how fast or how long it took us to come up with the Human Genome Project, right now we know about 54,000 proteins for humans. How long is it going to take to come up with the rest of the proteins? And considering what we know about the gene code and what we can do with it right now, how long until we can completely understand what all of these proteins are actually doing?
Well, you asked all kind of the global questions in proteomics. One, I would say, I think a lot of people feel there are going to be just 20-some thousand proteins, and that the other forms are modifications, splicing alternatives, editing, those kinds of changes. So there is a core set of 20-some thousand, and it's those we're going to create the MRM mass spec assays for. And we, as I said, we think we can do that in a few years. But if you get to questions of modification, of editing, of alternative RNA splicing, you obviously get into really complicated things that will take tens of years probably to really sort through, and a lot of it is just because chemistries aren't available for looking at a lot of modifications very effectively, although there are increasingly, there are some really very spectacular new studies on phosphorylation that I think is going to elevate it to kind of a global proteome level. But with the mass spec, you're always asking, what's the level that you can see? So in organs, there is about a 10 to the 6th dynamic range of proteins. MRM mass spectrometry can deal with organs really effectively. In the blood, there's a 10 to the 10th or 10 to the 11th dynamic range. It can't begin to deal with that unless you do front-end reduction in complexity and so forth. So I would see the Human Proteome Project that we're putting on as a phase thing where initially we'll focus on the human assays, then we'll bring in some of the classic model organisms and do their assays, and that's kind of the inverse of what we did in the Genome Project. And then we'll really move into the much more complicated questions of modification and splicing and alternative forms and spatial dimension, all the other things that are really important about proteins. So we hope we can convince federal agencies to do this over a 10-year period. It's not a very good time to be going hand in hand to to Congress and to the executive, but we're really going to push it.
Thanks.
So a big problem in neurodegenerative disease research that I noticed when I did a little bit of it was that the mouse models often don't recapitulate a lot of the things that happen in the human. And when you're going through this Prion example, it just occurred to me that maybe if you did something similar with the various transgenic mouse models, you could maybe ask whether or not the mouse just doesn't live long enough to manifest the symptoms, or whether or not there are whole sort of clusters of networks that are absent in the model. And I'm just wondering if anybody in systems biology is thinking about taking the approaches you described to try to understand the models themselves better.
So that's really a good point. I would say in cancer, we're beginning to get models now that mimic the human disease more and more closely, because they're mimicking the human defects more and more closely. What has been really interesting about Prion disease is the 3 downstream networks, glial activation and the generation of neural extremities and apoptosis. Those are networks that are seen in a lot of human neurodegenerative diseases. And I think if you deconvolute the problem from globally, does the mouse look like the human, to can we use particular networks as models of human disease, then I think you can start with things that aren't in close alignment and get a lot of information. But much better is being able to create models that mimic much more effectively the human disease. And that's really hard for complex diseases where you've got multi-gene dimensionality and things like that. We're, together with Mark and others, we're going to start looking at Parkinsonism, and that's one where I think there's really a good shot at beginning to mimic a lot of the human models, because at least we know a bunch of the genes. Now, I'm really worried we don't know the modifiers, and they could be, in the end, they really could be key. But if we use whole family genome sequencing in Parkinsonism, as we're going to do, then I think we ought to be able to get at the modifiers as well. But it's really a good point. But I think we'll be able to pick and choose if we deconvolute information to networks. And I think a lot of animal models will be interesting in particular features to their human counterparts.
So that was very inspiring, and I want to home in on one small point you made, which was that you predicted that the P4 approaches would be particularly cost-effective for, among other things, for developing countries. And I'd like to see how you envision that happening, because at this point it looks very, very technically intimidating.
Right, right. So I would, so I can give you 5 arguments that we could take, we could spend a half hour on. But let me just give you a thumbnail sketch of the arguments, then we can talk about any you'd be interested in afterwards. Number 1, I think diagnosis is going to transfer our ability to create an impedance match and stratification of disease against drugs, and I think one can be very, very effective in curing. And if you figure out how much it costs for colon cancer drugs and the 4 or 5% of the people that really respond, you can begin to see this kind of savings. So that's one point. Number 2, I think this whole idea of being able to use network approaches to identify targets that will, multiple targets that will let us manipulate network behavior is really going to be a powerful new way for drug target discovery, and I think it's going to change completely the dimensionality of costs in generating drugs. Now, you have to take some of that on faith. We're working in model systems to figure out how to do that. I think number 3, the whole idea of one of the 2 projects at The Ohio State is to set up parameters for measuring wellness. So in my view, each individual is surrounded by an envelope of wellness that has to be uniquely defined for each individual. And I think what those parameters will let us do is assess whether the vector of the individual is toward greater wellness or away from it, and to optimize behaviors as against wellness. And I'll tell you, you can make enormous economic arguments about how much money you save if you don't have people sick, especially important kinds of people. A fourth argument is the technologies that we're talking about now are changing exponentially in throughput and decreasing exponentially in cost. For example, I'm utterly convinced within 5 years we'll have a genome, we'll be able to do individual human genomes for $500 or so. And I think it isn't going to be any of the companies that you know and love out there now. The essence of what the new technology is going to have to be, for a variety of interesting reasons, is one, single molecule sequencing, and two, physical measurements rather than fluorescent measurements. Signal to noise is much better. And then the fifth argument I would make is I think there are a whole series of tools in medicine, and I'd say stem cells, I'd say, and areas like cancer, where I think there are really going to be some fundamental breakthroughs that will let us, again, deal much more effectively with those kinds of diseases in the future. So one of the key things that this P4 Medicine Institute is going to do is actually persuading a couple of really good economists to analyze these in detail, making reasonable assumptions, and see what the impact actually is going to be. Because that's the first question I always get asked. Prove it to me, because up till to date, everything seems to be getting more expensive. And really, it's getting more expensive because medicine's basically failed. We haven't figured out how to do medicine right. I mean, cancer therapy is the biggest disaster area that I can think of. I mean, for the money we spend and for what we get back, it's really a big disaster area. So.
Lee, lovely talk. In getting your list of prion response genes, were you able to intersect those with known susceptibility genes? Prion obviously is one of them, but there's several other mapped candidates and even strong candidates that should give you the causality from mouse models.
So we did, but when we have better mice strains like the collaborative cross, I think we can really do these experiments in a much more effective way. Because we just don't have a good way of looking at modifier genes now. And I mean, what I'm going to, there'll be a time when genomes will be $100 a piece, and it'll be easy to take the 1,000 different collaborative cross animals and sequence them completely, and it'll be enormously informative for the biology we want to do for those things. So we have done some correlations, and they're the kind of correlations you think, but I have the feeling it's just an iceberg. I think there are a whole series of really fascinating things we'll be able to look at.
Okay, well, I think that we should give Lee a